So what I'm going to discuss and share with you some of our work, and the mission that we are doing is the vaccine and therapeutic RNA for equity access. I think we learned a lot from the previous speaker that the threat globally is real, and the resources is getting limited. So I think each country have to work together, particularly the academic center and governmental funded organization, and together with the high income country as well. It's not going. So I like to get this quote. One of my friends that we work together for a decade now is so Drew Weissman from UPenn, who recently got a Nobel laureate, and he's one of the pioneer of RNA scientists. And what his quote is the issue is that although we have a very, very advanced technology and scientifically and biologically, but the low and middle income country remain left behind to access to life-saving and vaccine and therapeutics. So in our center, actually I found it with my team 2-3 decades ago, and recently formally called Chula Vaccine Research Center. And then we start initially we on the DNA vaccine, subunit protein vaccine, and 2 years before the pandemic started, we switched and started collaborating with Drew Weissman to mRNA platform. And we and Drew have a very clear direction that we committed to make the technology available widely. And we are a small team in the medical school, and the good news is that in the medical school you have a lot of expertise in different areas of diseases, and we can move, from the infectious disease to non-infectious disease very quickly, and particularly with this mRNA technology. We start, and I believe everyone's during the pandemics of COVID-19 started, no one really have, except Pfizer and Moderna, BioNTech, no one really have a good infrastructure of mRNA vaccine technology from R&D and moving to preclinical and to the clinical testing. But we have been proving that start from zero in 2017, and we started 2019. We'll be able to move from the lab and partner with Penn and also with the local manufacturer, eventually can bring the vaccine to the clinical trial, although it takes much longer time than BioNTech, Pfizer, and Moderna, because they have a lot of wealth and infrastructure and resources. And you can see that this is our Chula COVID-19 vaccine program, start in phase 1, and then because the process is too late, in a lot of things that we have learned, eventually we have to move from Wuhan strain into bivalent strain, and we also be able to prove that with that challenge we can make a bivalent vaccine together with the Wuhan and the Omicron virus. And you can see that we can make a phase 2 trial in Thailand, 450 volunteers, and the phase 1 in Australia, 1-2 in Australia, 150. And this is show you some of the results and showing that in green is our vaccine is called Comvigen, and in yellow is the Pfizer community bivalent vaccine. And you can see this is a 2-to-1 ratio trial. You can see that single dose boosting with the vaccine, either the comparator arm and the Chula vaccine arm, you can see that we got a non-inferiority results at 1 month after the immunization. Now, by the time we got this phase 2 clinical data, is mid of last year, so it's too late, right, to go for licensure and commercialized use. Anyhow, what we have learned is that now we be able to build up and settle, I would say, almost all the capacity that we can do. And you see that we can have an antigen design very quickly. Within a week, we can go to synthesize, and only the thing that we have to still to outsource is synthesize the DNA. And then we can do in vitro mRNA very quickly, less than a week, and then go to encapsulation. And can be tested in preclinical, in animal facility or in the non-human primates facility, also at National Primate Center at our university, and then can move to clinical development. The good news that I can share with you is that we also, because the hospital and the university realized that personalized medicine is very critical. So now we have a clean room facility to do both mRNA-based and then cell-based therapy. So we have that capacity to go for clinical lot production as well. Now, this is the pipeline of the vaccine. This is to be able to convince you that mRNA technology platform is really one of the options that can move things very, very fast. Now, during, just after pandemics, we now have more than 10 candidate vaccines in the pipeline. You see this is a viral vaccine, a bacterial vaccine, parasite vaccine, and a therapeutics vaccine as well. The one that in bold is the one that have a very convincing preclinical data or proof of the concept data, and some of them have been funded for clinical development. Now, this is just to highlight again that a number of listed that has been funded by the National Vaccine Institute Thailand. So this HPV, dengue, H influenza, H5N1, leptospirosis, tuberculosis. And this is funded by the Japan GHIT, the Global Health Innovation Research. And this is led by a group of Biotech experts in Mahidol University. Start from nobody in the past 5 years. Now we've been invited to join a number of key networks that hopefully we can contribute to make sure that we can make the equity access issue more feasible or possible. And here we start to we firstly joined Professor Drew Weissman initiated for global access since the beginning. And then we have been joined at the reading scientists of some of the vaccine development program by the WHO network, and also co-PI with the professor of Sheffield University, Professor Tak Sen Wong, to lead the UK South Asian Vaccine Hub. And then recently, we also had been invited to join Kiat Foundation RNA network through PAT coordinator. Now, this is the list of the 4 vaccine targeted, proposed by WHO MPP from that consortia. And you can see that dengue, HPV therapeutics, malaria VIVAC, and hand foot mouth disease. The highlight is these 2 have been well funded to go for clinical development by, this one is by Thai government, the NVI, and this one by the Japanese GHIT fund. Dengue, we also got funded by the National Vaccine Institute. Hand foot mouth disease, led by Professor Justin from in US. So far, we both didn't get any funding, so we use our seed money to start doing proof of the concept study. And this is a new facility that we have at the Chula Medical School. So the building called the Research and Innovation Building, RI building. At the 12th floor, this is a clean room facility. On the left-hand side, your left hand, is the CAR T-cell GMP facility. Now already start operating. This is the ex vivo CAR T-cell. So for treating leukemia and starting some of autoimmune disease to get up autoimmune B cell as well. On the right side is our facility that this is RNA. So we allow us by next year to start producing clinical lot mRNA products. I'd like to emphasize again that these two vaccines have been officially funded by the National Vaccine Institute Thailand to go for clinical lot production at our new facility and to go for phase 1 trial. And the avian flu H5N1 also has been funded. This vaccine right now, we already got into start producing the GMP plasmid grade. And the YVAX will be a year later already been funded by GHIT. I'd like to give you a brief information of our result, and you see that the threat is real. It's getting closer and closer. Last couple of months, we see more death cases from Cambodia as well. And this is the at least 11 cases total in Cambodia and 7 from in June and in July. So this is our candidate vaccine. So this is the standard immunogenicity study, immunized 2 doses, 2 weeks apart, and then collect the blood and the splenocyte for study. And the vaccine that we design is a multi-component or multivalent, either 1, 2, 3, 4, 5, 6, okay, 6 or 3 or 2 component. And you see that and even with a very low dose, 0.2 microgram. So when you're working on mRNA vaccine, in mice, if your vaccine can work no more than 1 micrograms dose per in mice, that is very likely to predict that it's very successful, highly to be successful in human. So we start with very low dose, and you see that although it's if you low dose, you're going to have a heterogeneous response, but it's significantly higher than the control group. This is again H5, and this again H7. And by including NA as well, and you see more and more T-cell response as well. And this is in process of the challenge in Follett at Cleveland Clinic, Ohio. The other one that I'd like to bring up to share with you is the therapeutic HPV RNA vaccine. So you look, this is the control arm. So this is the tumor size that you can, by imaging, you can pick it up by day 11. This is by naked eye, you probably on day 14. But you see this, the vaccinated group, you didn't see the tumor detected. And also we did the PCR of the HPV DNA, it's all disappear. And this is a tumor mass in the control arm and the vaccinated arm. Now, last but not least is that if you check ChatGPT right now, how many therapeutic RNA has been approved for commercially use is more than 10. And most of them either siRNA or in vivo CAR T cell, or in vivo CAR T cell is another, it's not yet there yet. But most of therapeutic RNA will cost up to $10,000 a year, or up to $500,000 a year. So equity access is very, very unlikely unless we start working on it seriously. So now we started working on siRNA, and then also start learning in vivo CAR T cell in the Drew Weissman lab. So I'd like to thank all the volunteers that have used some of data to share here, and collaboration between our campus, within Thailand and outside Thailand, and also all the PI for clinical trial. Thank you very much for your kind attention. Okay.